final sort of section that we're going to look at. So just a reminder about the nitrogenous bases, the nucleosides, and then the nucleotides. We're going to start putting them together because in the structures of DNA and RNA, we have a monophosphate on that nucleoside to give us that overall structure of DNA. However, when we start looking at the structures of DNA, we are interested in these base pairs here because certain base pairs hydrogen bond to the other side of the helix. So when we're taking a look at the structure of DNA, we have something called a double helix. So we have two strands. So rather than drawing them as a helix, I will just draw the strands. This would be the phosphate, the phosphosugar, or the phosphoribose or deoxyribose backbone. Okay, so that's always going to be the same. Remember that 3' to 5' over and over and over again. However, then we have those beta linkages to our nitrogenous base, nitrogenous base, nitrogenous base. Now, if you remember the double helix, there's two strands, but they sort of wrap around one another like a helical staircase. The nitrogenous bases face the inside, but the other thing is that you have 5' and a 3'. So remember, a free phosphate and a free OH group. The other strand is what we call anti-parallel. It goes in the opposite direction. So the base pairs face the inside of that helix. And so what you have is on your one strand, your nitrogenous base and your other nitrogenous base are held together with hydrogen bonds. So if you remember back from the general chemistry chapters, hydrogen bonds are actually not a bond. They are an intermolecular force. And that's important because in order to transcribe and translate our genes into proteins or other things that we may need, we have to separate those strands. Well, if they were covalently held together, it would take a lot of energy to pull them apart. But hydrogen bonds, you can easily pull apart and then have them come back together when you need to open the DNA to transcribe or translate a gene into protein or whatever it may be, okay? But you can also easily then put it back together. But here's the thing. There are only certain base pairs that go together, okay? For purposes of this course, the thing to remember is that A always in DNA pairs with T, or in the case of RNA, A always pairs with U. And the reason why I have two here is because there are two hydrogen bonds between an A pair and a T pair, as you can see on the slides to the left here. So we have A and we have T. So if you notice, we have a hydrogen bond donor and a hydrogen bond acceptor for each of those. So we have a pair, two hydrogen bonds for A and T. But when we have C to G, we have 3 hydrogen bonds. So we have 3 hydrogen bonds between cytosine and guanosine. Okay? That's important because it tells us about the strength of the interactions. 3 is more than 2. So we have more hydrogen bond interactions between C and G than we do through A and T. So that's also what we mean by the two strands are anti-parallel, as one goes 5' to 3' here, the other goes opposite, 5' to 3', so that they line up. Okay? So the strands are what we call also complementary to one another. So again, A pairs with T, C pairs with G. So here's the thing, if you know the sequence of one of the strands of DNA, you can already predict the other strand. So for example, if my one strand of DNA is A, T, G, A, G, you already know if this is let's say 5' and this is 3', well you already know over here, this is 3', and we know A always pairs with T, T always pairs with A, G always pairs with C, A with T, and G with C. And then we know that this would be my 5' end. So again, these strands are complementary. So you should be able to, now that you know how these pairings work, for every A, you get a T. For every T, you get an A. For every G, you get a C. For every C, you get a G. You should know if I give you the sequence of one, you should be able to predict the sequence of the other. Now remember, the only difference is this would be in DNA. If you're trying to predict the new sequence in RNA, all you do is it would be the same as matching here, except you would replace T's with U's. So if you looked at this sequence in RNA, it would be U, A, C, and then a U, U, C. So again, it's the same So if you're matching this strand, it would be the same exact as this strand. However, you just replace T's with U's. Because remember, DNA and RNA, same language, but slight differences. One of the differences, T versus U. So again, you should be able to, if you know the sequence of one of the strands of DNA, you should be able to predict the strands of the other piece. So here's the cool thing about DNA is every single piece of DNA, every single cell in your body that needs a copy of DNA, it's about 6 feet. Now imagine packing 6 feet into every single cell in your body. It's very difficult. So there's a lot of different types of packing that we do. So as you can see, you might have heard of chromosomes before. We have these kind of X kind of shaped pieces, if you may have seen pictures of them before. This is how humans pack their DNA. They're packed very, 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 very tightly, which means they need to be unwound to get at particular genes. So we go from our chromosome, we have what's called a nucleosome, because what we actually have is our DNA. To pack it very efficiently, we actually wrap DNA around a protein core. So we wrap this around very, very tightly. These proteins here, though, if you remember, these phosphates are negatively charged. So the proteins that are here are called histones. These histones are very positively charged so that they keep the DNA wrapped around them. So this concept where you have a histone plus DNA, okay, so you have a histone plus the DNA that's wrapped around it, that is called a nucleosome. So again, in the same way that we saw the nitrogenous base plus the plus the sugar plus the phosphate gives us from nucleoside to nucleotide. We can do the same thing here. A nucleosome is a piece of DNA wrapped around those histones. So that's where that comes into play. So again, we have those histones. And then what we can look at, maybe you've heard of like the Human Genome Project or getting your genome sequenced through 23andMe or Ancestry.com, things like this. Your genome basically is your entire sequence of DNA, therefore it gives you your genes. So this is just an example from your book, and please do not memorize any of this, but you can see like this is one particular chromosome, and it can show you where these particular genes are located and what maybe diseases or other markers they might be associated with. So you might have a BRCA1 gene, and oh, do you have the mutation, yes or no? You know, there are a lot of things that we can do by looking at the Human Genome Project and sequencing. It can tell you some information about what your history is in terms of what mutations you may have in your genome and things like this. That makes you more or less susceptible to certain diseases, more or less susceptible to certain characteristics that you may have, different things like this. So, and here's how this works. So the next set, the final set that we're going to look at is how do we replicate our DNA? Well, we already kind of know this because we know that if we know one sequence of one of those strands of DNA, we already know the sequence of the other. So if we have to go back to our central dogma of DNA to RNA to protein, we just have to read one copy of the DNA and we get the other copy. We know what we should be getting as a result of that. So when you actually take and you replicate cells, so if your cells are, dividing like your skin cells or your stomach cells, right, you're constantly losing cells, cells are dying, and you're making new copies of cells all the time, they need to have a new blueprint. So you need to replicate your DNA. And when we do this, to replicate your DNA, so you might have, if we go back to our central dogma, right, we go to DNA to RNA, but we might have to go DNA back to DNA to replicate itself. So this is just called DNA replication. So you want to make sure that you're definitely replicating very, very accurately. If you don't replicate your DNA accurately, that can lead to mutations and a bunch of other issues, right? So this is done by an enzyme called DNA polymerase. So if you remember back to our enzymes, anything that ends with ase is an enzyme. So this is an enzyme that makes a polymer of DNA. You can also have an enzyme that makes a polymer of RNA. The name of that is, guess what? RNA polymerase. So again, we name these based on kind of what they do. So when we replicate our DNA, remember one strand we use, because if we know that sequence, we can replicate the other strand and they run anti-parallel. This process, though, if we're replicating the DNA, we start out with the double strand, and we're going to make a total of another double strand for 2. But what we have is we're going to read each of those strands and then hook them up with the new strand. This is what we call semi-conservative replication, so semi meaning half. So we have half of the old DNA, and then half of it is brand new DNA. So if you take a look, we have here, you can see in your book from this picture, you have these brown strands, which are the original, but when you replicate them, the daughter strands are blue. So the new strands will have half of the parent strand and half of what we call the daughter strand, so the new strand. So what we call semi-conservative. Half of it is conserved, half of it is new. So if you take a look at those two strands, though, when we replicate one of them, and here's the thing about DNA polymerase, right? It can only read in one direction. So it has directionality. So we have what's called a leading strand. So we make that strand 5' to 3', and that just continues. So the RNA polymerase jumps on, and it just reads all the way through. However, the other strand, because it's in the opposite direction, DNA polymerase can only go 5 to 3, so it makes them in individual fragments. These are actually called Okazaki fragments. And Okazaki fragments are made on what we call the lagging strand because you have to make them individual pieces and then they get stitched together with DNA ligase. So there are a bunch of proteins. The main point is that there are a bunch of different proteins that come together to make and replicate your DNA. It's not just DNA polymerase that's required. So because of this, it's probably a good idea that we want to make sure that our DNA stays as accurate as possible when we're replicating it. Because if you were to replicate your DNA and every single time there was a mutation, that could have disastrous consequences. So we actually have about a little under 40% of all of the genes in us are there to make sure when we replicate our DNA and when we make a copy of it, everything is frankly, quote unquote, perfect or as perfect as it can be. So we have a lot of genes dedicated to the RNA polymerase, but also other genes and other proteins that go through and be like, hey, is this mutation supposed to be here? Hey, did you make a mistake? Double checking and proofreading. It's kind of like if you were turning in an assignment and you had 25 people who proofread your assignment for you before you handed it in. It's very, very good to do that because we know that different mutations can lead to huge and dire consequences. If you have an issue with the blueprint, building the building is everything from that point on will have an issue with it. So again, this is that flow of transcription and translation making that central dogma. If you have an issue with the DNA, you're going to have an issue with the RNA, you're going to have an issue with the protein. And none of that can be good. So we really, really want to make sure that the DNA is as proofread as possible. Okay? So this is the process of transcription is going to from DNA to RNA. Specifically, we use a particular type of RNA called mRNA. M in this case stands for messenger. It's the messenger that says, hey, this is the piece of RNA that we're going to send the message to make the protein. It's the internal message that we see that's going to give the message to the translator, aka the ribosome, to make the protein going from those different languages. There are other types of RNA, maybe you've heard of them. There's tRNA, transfer RNA, rRNA, which is a ribosomal RNA. That's part of the translator, okay, that you have. So your DNA is nice and packaged away in your nucleus to prevent any kind of harm coming through it. Your ribosomes are out in your cell and your cytosol. So they hook up with the mRNA and they can make protein. So remember, your mRNA is complementary to the piece of the DNA that you looked at. However, the difference being A, in your DNA goes to T in your DNA, but it goes to U in your RNA. Okay? So remember, transcription, same language, slight differences. One of being T's are replaced with U's. That's how our body can differentiate. Is this a piece of DNA or is this a piece of RNA? And that's important. So remember, the mRNA is complementary, right? A always pairs with U, T always pairs with A, G always pairs with C, and C always pairs with G between DNA and RNA. So if we take a look at that transcription, right, to make the messenger RNA, so remember, replicating DNA to make more DNA is DNA polymerase. Taking DNA to make a polymer of RNA, right? So DNA to RNA, this is done by the enzyme RNA polymerase. You're making a polymer of RNA. So it's very similar to DNA, but we don't actually have as much proofreaders for this process as we do for DNA replication. Because remember, RNA actually is not as stable because you have that OH group on there. And so as a result, we're not as vested and interested in making sure that our RNA stays as faithful as our blueprint, right? If you make a mistake and you bring in 30 chairs instead of 29 chairs into a room, you can just redo that again, right? You can just take out a chair and everything is good. But if you had an issue with the blueprint itself that told you, oh, this room needs to have 50 chairs, but it can really only hold 30 chairs, that's an issue with the blueprint, and that's a bigger issue. So again, the piece of mRNA, right, is complementary to what we're talking about here, as you can see in the example, this picture from your book in the slides. You can see here we have what's called the coding strand or the template strand. So again, the mRNA transcript is complementary to the template strand, but it is the quote-unquote same as the coding strand, just replacing T's with U's. So in this case, this sequence here is the coding strand. This sequence here is the template strand. So template strand is complementary to the coding strand or the piece of mRNA transcript that we have. So finally, we go now from RNA to protein. So when we go from RNA to protein, remember we're changing languages now. So we need a translator. So this translation that we have, right, is done by the ribosome and transfer RNAs. So this is where we're going from the language of nucleotides to the language of amino acids. So the translators that we use are what we call codons. So we look for 3 nucleotides at a piece. Each 3 nucleotides codes for 1 amino acid. And so we have, and please do not memorize this, but there are certain nucleotides, and we know what we call the nucleotide code. So we know the codon, there's like a codon table, which we'll see in the next slide, where we know these 3 nucleotides code for this amino acid, okay? But here's the thing. Based on the number of codons that we can have up to 64 different codons. But if you remember, we only have 20 amino acids. So there is what we call redundancy in this code. So some codons code for the same amino acid. For example, if you're taking a look at the slides here, you can see ACU, ACC, and ACA, and ACG all code for threonine. So if you actually, quote unquote, have a mistake and you accidentally, instead of putting ACC, you've made ACA, it still codes for the same amino acid. So that's actually kind of great. So this code is what we call redundant. So again, you should be able to look up this code and you should be able to figure out what amino acid is coded for what codon, but please do not memorize it, right? So if you know that your first nucleotide is A, your second is U, and then your third is G, you have AUG, that codes for methionine. If you know your codon is GGG, you look G, G, and G, that codes for glycine. So again, you should know that there's 3 nucleotides per 1 amino acid. That's where we get this genetic code. That's where we're going from our nucleotide language to our amino acid language to make our proteins. And hopefully our proteins, right, with the correct structure, we get the correct function looking at the primary, secondary, tertiary, and quaternary structures. So again, looking at these, again, our translators are the ribosome and what we call tRNAs. tRNAs are a piece of RNA that attaches to the amino acid, so it physically brings the amino acid to the ribosome to start making that peptide by creating peptide bonds. So if you look, the tRNA has to match or be the anti-codon to that. So if you're looking for a codon that starts UAC, the anti-codon would be AUG. And that's what the tRNA has. So that's why this whole complementary strand, template strand, again, that's how you can read those, is because once you know one sequence, you automatically know the other, okay? So this translates between the nucleotide language, the codon language at the bottom, for example, of your codon in your slides here, and it's hooked up, only certain tRNAs are hooked up to certain amino acids. So if you match the anti-codon, if your codon matches the anti-codon, you know this is the amino acid. So if you have an issue with your tRNAs, your translator's getting screwed up. It would be like, hey, I have this particular translator, even though they're supposed to be speaking French, they're speaking Italian or something like this. That can be an issue. So again, these translate between the codon, so they have both a codon, and then they have an ester linkage between the tRNA or the piece of RNA and the amino acid. The ribosome is going to take that amino acid when that comes into the ribosome and make the peptide bond between taking off that amino acid and putting it onto the existing other amino acids to start creating the protein. So again, without getting too much into the weeds on the the ribosome. The ribosome is this big, big, complicated piece of machinery. It's got protein and it's got RNA because, right, so we have to read RNA. So there's a point where it comes in, the tRNAs come in, they bring the amino acids, a whole bunch of stuff is there. So you've got all of these RNAs kind of colliding in the ribosome, plus a bunch of protein. So you've got the mRNA coming in, you've got the tRNAs coming in, and then you've got the ribosome needing to read some of that information with these rRNAs. So you've got a lot of these things, plus you've got protein in there that's an enzyme. So the ribosome is actually a complex, but there's what we call peptidyltransferase, ACE for enzyme, activity to start making those peptide bonds to grow that protein. So it's a large complex structure of protein, different types of RNAs in there to basically make the protein from the mRNA piece that you have. So here's the thing though, in your genetic code, right, I've been focusing on this, the DNA needs to be as accurate as possible. That blueprint needs to be accurate. If you have mutations in that DNA, it can lead to what we call missense mutations. So what that can mean is instead of an alanine, I accidentally put in a serine. Now sometimes that might not be a big deal, or in the case of like sickle cell anemia, you replace, right, a glutamate with a valine, and it causes a huge issue. It causes a disease as a result of it, a pretty catastrophic disease. So some amino acids, because of that redundancy in the code, right, GGG and GGA both code for glycine. So if you accidentally have a mutation in your DNA of GGG instead of GGA, you'll still put glycine in there. So your protein actually will be exactly the same. So that's not an issue. That's what we call a silent mutation. You put in one amino acid for the same amino acid because of that redundancy in the codon code that you have. But then there are some proteins or some mutations where you actually get what's called a missense mutation. I change one of the codons or one of the base pairs in the codons, and now I get a different amino acid, and that can not really change much, or it might completely derail the structure and therefore function of the protein. Then we finally also have what are called frame shift mutations. This is if you insert a nucleotide accidentally or delete one accidentally or two. That can have catastrophic consequences because then you're shifting everything up. So imagine you're reading a book, but they accidentally transposed and they just put a space in between a word, and then they just kept the same number of letters for each subsequent word. It would be really, really hard to read. You'd get a completely different type of book as a result. So again, it changes all the downstream amino acids usually as a result. That's a big issue. Okay. So overall, I would like to just very much thank you for joining me on our journey through biochemistry. If you have questions on carbohydrates, lipids, proteins, or nucleic acids, please let us know. Thank you very much.